Okay. Thanks for the introduction, and I wish we could all be there to do this in person, but so be it. What I'd like to visit about is a project that I've been working on that combines a couple things that we've looked at in the past few years. Both the low-pass sequencing and some of our work on cow weight and productivity. If I can make these slides advance. Okay, so, the low-pass sequencing, we've explained it in quite a bit of depth earlier, but, what it boils down to is low coverage whole genome sequence that's imputed to a whole set of variants a large set of variants that are detected in an imputation reference panel. And this can be similar or lower cost than SNP arrays, and we get much more comprehensive genotypes. And some recent work shows that the imputed genotypes can be more accurate than array genotypes called from the array. This panel up here is actually from human data where they compared imputed genotypes imputed from different low pass and genotypes called from a SNP array to the genotypes that were obtained from deep sequence, about 30x sequence, where in all cases the imputed genotypes were more accurate than the array genotypes. We haven't done that exact comparison in cattle, but what we have shown is that we can get high accuracies in comparison to what was called from deep sequence, up around 98 to 99 with the little, at the lowest allele frequencies, we're not quite as accurate as higher frequencies, but we're still doing quite well. And we've also did some work on some cow productivity traits, different using random regression models with cumulative traits. And for this work, I'm just focusing on weight weaned, where it's the total weight that a cow weans over several years. And in this case, we're not adjusting for calf age because we want to favor cows who consistently conceive and calve earlier than their contemporaries. So they'll wean older, heavier calves. And for cow weight, we're just looking at the weight measured when we preg check. And with the random regression, we can get a record every year and then project that out to any age where we do have records. For both of these traits, there's strong correlations among ages within traits, and the traits are weak. There's a weak negative correlation between weight weaned and cow weight for most ages. But so our objectives with this were to estimate additive and dominance variance in cumulative weight weaned and cow weight that we can attribute to functional variant genotypes that were imputed from low pass sequence, and then characterize animals by their predicted genomic merit. And for the observed females, we're looking at both additive and dominance effects on the genomic prediction. The data we used were all from the germplasm evaluation project, where we've so far genotyped almost 22,000 animals with one array or another. And, so though combining all of the arrays, we've got genotypes for over 900,000 different variants, mostly on the bovine HD chip and the GGP F250. And we also had low pass sequence on about 3,000 animals. Half of those were also genotyped with an array, and half are just have the low pass. And there were, in that run of the low pass, we got genotypes for about 60 million variants. And a lot of the animals genotyped with that were dams. But between the two approaches to genotyping, we have over 22,000 animals with some form of genotype. And to impute those to the genotype GPE, we did 2 steps of imputation. First, to impute all the array genotypes to the combination of the bovine HD and F250, and extracted those variants plus variants that were interesting according to the annotations. So that was over 1 million protein variants in protein coding sequence, untranslated regions, and non-coding RNA that might affect regulation. And put those two, the low pass and the imputed arrayed genotypes, together in another set of round of imputation to impute the low pass genotypes to the full set of animals, and then removed the uninteresting chip genotypes from that set. So we wound up with almost 1 million functional variant genotypes that we used for these analyses, and built both additive and dominance genomic relationship matrices from that set of genotypes. And the cow records we used were also from the GP, or cows that genotype GPE cows. For weight weaned, we created a record for each breeding season that a female was exposed, starting when she was bred as a yearling to calve as a 2-year-old. And that weight was accumulated for each breeding season. If she didn't calve, then her record for that breeding was, if she didn't wean a calf, the record for that breeding was the same as her previous record. If she was pregnant when she was culled, that record wasn't used because she didn't have an opportunity to wean that calf. And I also had a term for cumulative calf sex that was total number of males she had weaned minus the number of females, to account for sex difference in weight weaned. So there are about 6,000 females with records. Most of them they averaged about a little over 5 records per female. And the pregnancy test weight was just recorded when we at the preg check following each breeding season. You know, we did the analysis with wombat and a random regression model with wombat. The effects in the model included cow age in half-year increments that allowed for open cows slipping from spring to fall or fall to spring. And the contemporary groups were birth year, season, and their yearling mating group, and assuming that all females in that mating group would be treated the same and have the same opportunity for lifetime production. Random effects included the genomic additive and dominance effects and the permanent environmental effect. And after a couple months of iterating on that, to characterize the animals, once we got the predictions, we used the pedigree-based breed composition and heterosis calculations. You know, after months of iterating on the variance components, we finally got it to sort of converge. So, you know, the heritability estimates were similar to what we had seen with the pedigree analysis. The cow weight heritability is high and fairly constant, around 0.5. The weight weaned heritability is near zero at age 2 and increases with age up to about 0.15 around from 8 to 10 years old. The dominance is negligible for cow weight and up until about age 8 when it starts to increase, and after age 3, dominance is about double the heritability on weight weaned. And this slide shows the distribution of both the weight weaned and cow weight predictions, you know, the sum of their additive effect and dominance effect for each cow. You know, there's a little over 700-pound difference in 8-year-old weight, cow weight, and 1,700-pound difference in weight weaned. And the scatter plot is color coded by breed. And from this, all breeds are contributing. There's a lot of overlap among breeds, and all breeds contribute to each of the 4 quadrants I've divided by the mean value for going from low cow weight weaned, low weight weaned, low cow weight, and then, you know, low cow weight, high weight weaned, high cow weight, high weight weaned, and high low. And to look closer at each of those quadrants, I've included the scatter plot and color coded it by what we're looking at, so that the breeds in blue are overrepresented in the blue half of in this case, the blue half of the plot, and the breeds in red are overrepresented in the red half of the plot. You know, so there are some differences for cow weight as well as weight weaned, where either above or below the mean weight weaned. And then for each of the quadrants, we also see differences in what breeds contribute more to those, to either in this quadrant or in the other three. This is starting from going around from the low to the high weight weaned, or low cow weight, high weight weaned, then the high, and high low. And also looked at the, both maximum breed content regardless of breed, where in most cases, most of these cows are half of the cows are between 50% and 75% of any one breed. And where we see the differences are, in either the high weight weaned group or the quadrants with high weight weaned. That, those cows are almost, the mean of the high weight weaned cows is little over 50%, and half of them are, and we don't see many high weight weaned cows that are, approaching purebred status of any one breed. And the heterosis for those cows is also quite different. That, the median heterosis for the high weight weaned is close to F1, where most of the other groups have lower pedigree-based heterosis. But all the everything with the high weight weaned has that high heterosis that we don't see in the other groups. So just to try and summarize this, the additive heritability estimates are similar to what we saw with the pedigree analysis, where the cow weight heritability is around 50% across all ages, and for weight weaned increases from near zero to about 15% at older ages. And dominance doesn't appear to have much effect on cow weight, but is extremely important for weight weaned. For all the current GPE breeds are represented in all the groups of cows split by the mean 8-year-old weight and weight weaned, but there are some differences in what breeds contribute more or less to each of those groups. But the heterosis is similar for the high and low cow weight groups, but the high weight weaned cows have higher heterosis than the low weight weaned. There's still a lot left to do on this. Where we have, with GPE, we have extensive pedigree records that we can use for breed composition and heterosis, but for other cattle that may not have that those pedigree records, we can estimate breed contributions from the genotypes, and also work on heterosis or heterozygosity with the from the genotypes as well. And I still haven't estimated individual variant effects, either additive allele effects or genotype effects from the dominance. You know, I was interested in our first talk today where we saw non-additive effects on bull fertility, and I'm hoping to see the same sort of thing when we do this estimation, when we estimate the effects on the cow productivity. And we need to work probably, just given the effects of heterosis or dominance coming out of these analysis, we need to do some work on mating strategies to, you know, maximize heterosis or the favorable functional variant genotypes. You know, for cow weight, looks like we can control that pretty well with just additive EBV, you know, especially if we had a crossbreed mature weight EBV, so you could select from different breeds with the same genetic potential, and then work on the breeding strategies for heterosis. And you know, this wound up with a lot of people involved. From all of the low pass genotypes came from a collaboration with UNL and Neogen, and the group at GenCove is behind all the imputation from low coverage sequence to the reference panel that they've put together. A lot of the computing was done on the UNL clusters in the Holland Computing Center, and I can't name everyone at Clay Center that was involved in collecting data and doing the tissue sampling and repository and the sequencing that we do there. So with that, I'll try to answer any questions you might have.
Questions from the audience? I'm going to start with one. All right, Dr. Snelling, can you, and I may have just missed this, can you better describe the pregnancy testing weight of those cows? Was that off of AI, off of natural breeding, 30 days post using ultrasound? What went into that measure?
I'm not hearing you well.
All right, I'll try to repeat it. I may have missed it in the presentation, but can you better describe the pregnancy testing weight of cow variable you had? Was that based off of AI, natural service, 30 days post, via ultrasound, or what protocol did you use?
Okay, you're asking about the pregnancy test?
Yeah, pregnancy test weight.
Okay, it was, okay, it would have been after the 60-day breeding season, and we just take away when the cows run through the chute.
Right, 60 days is probably what I was probably trying to get to there. Hopefully you can hear this question better. What is the magical number of calls or imputations to equate to a 50K or a 250K for those traditional cattlemen that have been pulling 50Ks or 250Ks based on your low pass research?
But let me try a different connection on this.
What is the magical number of calls or imputations that you would say to equate to a 50K or 250K that our traditional breeder is out collecting today on SNPs? I looked
Okay.
at that one slide and it was pretty high. Looked like.
Okay, we're from the low pass. We're getting 60 million genotypes called.
So in terms of cost, how comparable is that cost to a 50K, for example?
And that cost was, I think, the same as the GGP 100K.
Okay. I don't see any more questions. So I'll ask one more if I can. I'm curious about the low cow weight, high weaned calf weight. Was there a difference in heterozygosity and or homozygosity in those cows that excelled? I would assume there's a lot of heterozygosity there.
Yes.
At what percentage would you think in that particular call would we see? Is that 80%, 20%? I'm just not sure.
Okay, those cows were almost all 50-50 F1 cross. And I haven't looked at heterozygosity from the genotype calls yet.
Okay. All right, no other questions. Let's give Dr. Snelling a round of applause, please. Thank you, Dr. Snelling. We appreciate you taking your time this afternoon to be with us.